Hello everyone, this is Unit 3, Part 2, all about cells. Last PowerPoint we talked about the organelles and the cell membrane. This time we're going to talk about DNA, mitosis, and how proteins are made, as well as a little bit about cancer and cell differentiation. So as you know, inside all cells is the nucleus. Some cells are only, like red blood cells, are only formed with a nucleus, and the nucleus then is expelled. So red blood cells don't have a nuclei, and some cells have multiple nuclei, like muscle cells, for example. But all cells originally in the human body have a nucleus, and inside the nucleus is the DNA. The nucleus itself is made up of phospholipids, just like the cell membrane. Sometimes it's referred to as the nuclear envelope. There are large pores in the nuclear envelope that allows things to move in and out, so it's not quite as selectively permeable as the outside cell membrane. Inside the nucleus is the DNA. The DNA is in the form of chromatin, which is long, thin strands of DNA. Only when it gets condensed into short, thicker bodies of DNA, I guess, is it called chromosomes. But chromatin and chromosomes are both forms of DNA. But in a cell that's just hanging out and doing its normal job, the DNA is in the form of chromatin. So here's a picture of the nucleus, and it shows a chromosome, and if you stretch that out, it's actually coiled up chromatin, and if you uncoil it, the DNA is wound up around little proteins, those little yellow discs in the picture called histones, and when you remove those, then you can see the double helix. The double helix is like a ladder that's twisted, and that's probably the image you've seen when somebody says, oh, here's some DNA. But inside cells, it is coiled around histones, and then those are coiled up around like an old phone cord. If you stretched out chromatin, it would be like 6 feet long or something, or 20 feet. It's really long, and of course our cells are really, really tiny, so packaging of DNA uses histones to coil it up and to fit it in the nucleus. If you look at the double helix, though, you can see the blue bands here are the sides of the ladder, and that is made up of alternating sugar and a phosphate group. The sugar is deoxyribose sugar, which is the name, deoxyribonucleic acid, where that comes from. And the sugar phosphates are hooked together covalently, and hooked to the sugar is a nucleotide. Remember, nucleotides are the bases, or are the building blocks of the macromolecule DNA, or nucleic acids. And if the sugar on one side of the ladder is attached to an adenine, abbreviated A, then it is hydrogen bonded to the T that's covalently attached to the other side of the ladder. So if there's A on one side, you automatically are going to have a T on the other. They are complementary base pairing between the A's and the T's. A and T are always together. G and C make a good couple. They are always paired together as well. But everything is covalently bonded except the A and the T. The rungs of the ladder are held together by a hydrogen bond, which, as you remember, are much weaker, and heat can break them up. So if you heat DNA, it actually separates, the sides of the ladder separate from each other. So when DNA replicates, if you have a piece of DNA like the picture on the left here, okay, when the cell you need to make more cells, you need to replicate the DNA. So you take this 2 strands of DNA, and the cell has an enzyme that opens it up. It was creatively called unwindase when it was first discovered. And then an enzyme that adds new nucleotides called DNA polymerase reads the nucleotide that's left over there and adds a new one. So the blue in the picture is the original, and the new nucleotides in red are added. And of course you know what to add, because if you have ATC, then you're going to add TAG. Always A and T are always paired together, and G and C are always paired together. So the ladder simply splits in half lengthwise, and a copy is made of each side, and at the end you have two exact copies of the original, where half the ladder is the original DNA and half the ladder, in red here, is the new DNA. So when does a cell need to replicate? Most of the time, a cell is just doing its job. Your muscle cells are contracting and your skin cells are protecting you, and your stomach lining cells are producing acid, etc. But when there's damage or growth needs to occur, the cells need to replicate. And in this picture is a picture of an overall cell cycle. Growth and normal metabolic activities is referred to as the G1 phase. And again, most cells stay there until they are induced to replicate because growth, injury, etc. So that red arrow, some sort of inductive process occurs, or something says, hey, we need some more of you. The DNA replicates in the S phase. There's a pause where things are checked and you get prepared for cell division, called G2, and then you go into what's called mitosis. Mitosis is when the cell actually goes from one cell to two identical cells. And there are 4 stages of mitosis: prophase, metaphase, anaphase, telophase. And at the end, you end up with 2 identical daughter cells to the original. Same DNA, they both have the same organelles, etc. So here's a picture of the stages of mitosis. During prophase, the nuclear membrane breaks down. The DNA has already doubled, so it pairs up, and a spindle is formed out of microtubules. Basically, the chromosomes, which are now short condensed chromosomes instead of chromatin, so you can see them when you stain them under a microscope and look at them under a microscope, attached to that spindle. When they all line up in the middle, that's called metaphase, and then they split. Half the DNA goes one way, half goes the other, and when they move away from each other, these sister chromatids, it's called anaphase. And when they get to the opposite ends, a new nuclear membrane forms around each set of DNA, the spindle disintegrates, and the cell pinches in two. That is telophase. Okay, so prophase, metaphase, anaphase, telophase are the 4 stages of mitosis. To get a closer look at one of these chromosomes, it's 2 sister chromatids attached by the centromere, and the centromere is also what's going to attach to the spindle. Chromosomes, you have 23 pairs of them, one from mom, one from dad, contain genes, and each gene codes for at least one protein. And we're going to talk about that in a few minutes here. A little bit more about mitosis. Mitosis can happen very, very rapidly or very, very slowly. Your red blood or your bone marrow is making red blood cells at a few million per second. So it's happening very rapidly right now, all the time, because your red blood cells only last about 3 months, maybe 4, and they get too raggedy and you need more of them. Your skin is reproducing all the time because as dead cells get sloughed off when you shower or wash or rub your hands against each other, new cells underneath are being produced and pushed up. What makes cells reproduce? We know that injury does, growth hormone, or when you grow, that causes it. We also know that certain things stop cell reproduction. You don't need new cells, then there are certain molecules that show up in cells and reproduction is stopped. The problem, or one of the reasons we study this, though, is when reproduction can't be stopped or goes uncontrollably. Essentially, uncontrolled cell reproduction or uncontrolled mitosis is cancer. And we know about certain genes that can become mutated that cause mitosis to keep going. Proto-oncogenes are genes that turn on mitosis, and if they're broken and stuck on, like your car accelerator is stuck on the floor, you get uncontrolled mitosis. We know we have tumor suppressor genes that stop mitosis, and when they get broken, you have uncontrolled mitosis. So that would be like your brakes failing. When your accelerator is stuck on the floor and your brakes fail, you're really in trouble. And that's the same is true with cells that have both of those mutated, basically replicate uncontrollably, and that can lead to cancer. So scientists are very much interested in what induces and what stops and what triggers mitosis, because if we could figure that out, maybe we could control it better and treat cancer better or more quickly. We talk about cancer again in AMP2, but the National Cancer Institute, the link is here if you're interested, has a really good summary of what they're doing and different kinds of studies, as well as different treatments for cancer. Okay, so cells, when conception happens, you are one cell big, and mitosis then occurs to go from 2 cells to 4 to 8 to 16, etc., until the 70 trillion that you are today. But as cells replicate, at first, every cell has the potential of being anything. When you're one cell big, that cell has the potential to become any kind of cell. 2 cells, if you split them apart, can still become 2 individual identical human beings. When you're 4 cell big, each one of those 4 can become an identical human. But by the time you get to 8 cells big, you cannot pull one of those out and get a human. Already there's a head and a tail, or there's a little bit of specialization. And as you develop, the cells become more and more specialized in their function, and that's called differentiation. And the further a cell differentiates, the less it can go back. It can't return to what it was before. Once you get to be a muscle cell, you're a muscle cell. You cannot be a fat cell or a skin cell or a neuron. You are fully differentiated. Stem cells are the cells that have potential to become different types of cells. And during development, that's the week 2 of development, you have 3 basically types of cells: endodermal, mesodermal, and ectodermal. And each can become different things, but mesodermal cells can only become muscle, fat, bone, and cardiac tissue. And the ectoderm can only become skin and nervous tissue, for example. And stem cells and stem cell research is a big deal, because maybe we could make new pancreas cells for people who are diabetic, for example, from the endodermal line. Anyway, the further differentiated you get, the less likely you can go back, and the cells become very specialized in function. So it's too late once you become a lung cell or a heart cell to turn around and become a pancreas cell or a brain cell. What else DNA does is it directs the production of proteins. Remember, the unit of DNA that codes for a protein is called a gene, and they figure we have between 20 and 25,000 genes, and each one of those codes for at least one protein, some more than that, and the collection of proteins that you have give you your traits, make you who you are. Okay, so lots of us produce the same most of us produce the same proteins all the time. We all produce insulin, for example. We produce actin and myosin for our muscles. We all produce keratin. But at some time during development, for example, protein was made that caused my bones to grow, and maybe I didn't have quite as much of it because my bones didn't grow very much and I ended up short. Whereas my brother, who is 6'1", must have had more of it because he's almost a foot taller than me. So the protein amount and when it is there, and maybe the how long it lasts during certain times, determines the differences between people. 99.9% of the proteins in our bodies match everybody else's. But some people make a little bit more melanin and their skin is darker. Some people had different proteins around and their cheekbones form different, so we have different shaped faces, for example. To go from DNA, the directions in the nucleus to making proteins is called the central dogma of cell biology, and it involves two processes, transcription and translation. DNA is transcribed or undergoes the process of transcription to create mRNA. mRNA is used in translation to produce a protein. Okay? And again, it's the proteins that make each one of us unique. The RNA, which we haven't talked about yet, is kind of the middle step here. It is a single strand of nucleic acid material rather than double, and it has the letters A's, G's, C, and U instead of T. Uracil substitutes for thymine in RNA. So I'll talk a little bit about the RNA, and then we'll come back to this transcription and translation. The sugar is different, it's single-stranded, and the nucleotides are A's, G's, C's, and U's. That's how it differs from DNA. Okay? DNA is double-stranded, has deoxyribose sugar, and has the nucleotides A's, G's, C, and T. But RNA is single-stranded, has a ribose sugar, and has the nucleotides A's, G's, C, and U. There are three main types of RNA. And the mRNA, the messenger RNA, is the one that is going to code for a protein. And that's really the one we care about and we'll talk about the most. There are two other types of RNA that all our cells make. tRNA. The T stands for transfer, and it is going to be carrying or transferring the amino acid to the ribosome. And rRNA, which makes up ribosomes. Hopefully you remember that ribosomes are where protein synthesis takes place. And hopefully you remember that amino acids are the building blocks of proteins. Okay, you should have remembered that from the last two lectures. So make sure you keep that in mind. Okay, and as we go through, we're going to talk about transcription first. Some of that stuff hopefully will come back into your head, right? All right, so central dogma of cell biology says DNA to RNA is transcription and RNA to protein is translation. You need both of those to happen, transcription first, then translation, in order to make a protein. Step one is transcription, and it is the synthesis of RNA from a strand of DNA. One thing that's important to remember is the DNA is not turned into RNA. Okay, it doesn't magically convert to something. DNA is just used to synthesize a piece of RNA. Okay, so in this picture, the DNA is those two blue strands with the yellow between it, and an enzyme called RNA polymerase comes along, and it finds where the gene starts. Okay, DNA replication, you got to copy the whole 3 billion letters, but in transcription, you're only going to use the RNA polymerase to read the DNA wherever the gene is coded for. And a gene might be 1,000 base pairs long. Okay, it might be a short little section of the whole DNA. And it reads the DNA and it generates a piece of messenger RNA, A's, G's, C's, and U's hooked together, that matches the top strand of the DNA, except all the T's are U. Okay, the thymine is substituted by uracil in the RNA. So if you look at the bottom picture there, the DNA is in blue, and the top strand is A, T, G, A, T, C, and the RNA polymerase basically creates A, U, G, A, U, C, exactly matches that top strand, except all the T's are substituted by U's, because it's reading the bottom one and adding the complement. Anyway, what you should remember from this is transcription is the synthesis of RNA from a DNA template. Okay, so the first step to making this protein is transcription. So this piece of mRNA is created in the nucleus because that's where the DNA is located, right? And it goes out of one of those nuclear pores and it finds a ribosome. Remember, ribosomes are where protein synthesis takes place. The ribosome is two subunits. Looks like a great kind of a teal-colored blob in the picture. And the piece of RNA, the mRNA, is pushed through the ribosome and it's read 3 letters at a time. The ribosome looks at 3 letters. Those 3 letters are called a codon. And when it sees, like this one here, ACG, when it sees that one right there, a tRNA with the matching anticodon drops off whatever amino acid is coded for by ACG. Remember, tRNA is the transfer RNA, and it's transferring an amino acid to the ribosome. Okay, so the amino acids are floating around in your cell because you had protein for dinner, and protein is broken down to amino acids by the digestive system. And now we're going to build back a protein that your body needs by taking those amino acids and hooking them together in whatever order the mRNA says to. So translation is the synthesis of a protein, and it uses mRNA as the directions. It uses the ribosome as the factory, that's the place where it happens. It uses tRNA to bring the amino acid into the ribosome to attach. Obviously you need amino acids, so you need to have eaten for this to work. And when you hook amino acids together, you're creating a peptide bond, and that takes energy. Remember, energy in a cell is ATP, and we talked about how that is generated in the last PowerPoint. Okay, so check this link out. It shows a really good, quick few minutes on how protein synthesis happens, transcription and translation. If you go back to that picture here, at the end you're going to get this string of amino acids. It's going to fold up and become a functional protein. Another way to think about this is that DNA is like a recipe for a cake, and the protein is the cake, right? You can't eat the recipe, but you can use it over and over and over again to make more cake. And that's exactly what happens in your cells. The DNA is sitting there. Anytime you need more of that protein, you can do transcription and translation, and voila, you have more cake. Okay, so when you eat DNA, does it hurt you? Does it change you? Because when you eat bananas, you're eating A's, G's, C's, and T's. You're eating banana DNA. And no, it doesn't hurt you. You digest it just like anything else. And those nucleotides, A's, G's, C's, and T's, can be used to make more DNA for you. One thing cool about DNA is A's, G's, C's, and T's are the same in bacteria, giraffes, tulips, pine trees, and you. Okay, and you eat DNA all the time when you eat cells of other organisms. What happens when the DNA gets mutated? When you change the order of the letters, you change the order of the amino acids in the protein, and now the cake tastes different. You have a different recipe. Or maybe the cake's not made at all. Or maybe you make too much cake. Any of those kinds of things can cause a problem in cells. Okay? If transcription and translation didn't work, you wouldn't be able to make new proteins, and that would eventually probably kill you, because that cell wouldn't be able to make any enzymes or any transporters or anything else that it might need to survive. All critters on Earth, from plants to mushrooms to bacteria, have the same DNA. We can now move around genes, including putting some genes into other organisms, like creating GMO food. It doesn't hurt us to eat this, it's just more letters, A's, G's, C's, and T's. So it's not dangerous in the sense that we can't eat them. They do worry about some other issues with GMO foods. Is that good? Is it bad? If we can fix it, okay, they just okayed, the FDA approved a gene fix-it procedure to fix hemophilia. Hemophilia is caused by a one-letter change in the gene for hemoglobin. You guys remember that hemoglobin is a protein, or maybe we haven't talked about that, is the protein that binds oxygen in red blood cells. And if we can fix it, should we be able to, that would be pretty cool. But what if we could fix other things? That is the end of Unit 3. Again, make sure you read your reading assignment, check out Canvas for other links, and you can always email me.